我们就即将今天十一大学的医学院大师传习系列的第11场哈。那我们今天是采用现场跟线上的混合式。那我们今天的主讲者是刘扶东院士，他人现在是在我们台北哈。那我们同时有一些老师同学是在线上。今天一开始是由刘扶东院士来进行演讲，那大概时间会有差不多60分钟，然后还会有10分钟左右的 Q&A。那接下来会有一个 coffee break。那接下来是另外一个重头戏，是我们有我们的这个 PhD student， 他们会做他们的一个所谓的brief 的 introduction for their research， 然后呢再请刘院士给他们一些指导。好，那这是我们今天整个的流程。那我们现在要把它 switch 到那个英文版的哈。Ladies and gentlemen, on behalf of the Vice Dean of the College of Medicine, Tzu University, it's a great pleasure for us to welcome all of you to attend this talk by Fudong Liu, Academia. Professor Liu is the chair professor in Tzu University. And let me introduce Professor Liu briefly. Professor Liu, in the beginning, graduated from the Department of the Chemistry. So he also is a chemist. But after that, he went to the US, to the University of Chicago to get his PhD in chemistry, and also get the medical doctor degree, the MD in Miami University. And Professor Liu is an outstanding researcher in chemistry, biochemistry, and even in the immunology. And Professor Liu, right now, he is the vice president in Taiwan Sinica Academica, and also is a distinguished professor in a lot of the university. I hope your active participation in Professor Liu's speech will inspire your future research topics or approaching a study. And enjoy this talk, and please provide a warm welcome to Professor Liu.
Thank you. I want to start with thanking Dean Chen for your invitation, and also thank VP Xie for your kind introduction. So it's really a pleasure to visit Tzu University Medical School again, to albeit is again online, to share with you my research. So I gave my research is mainly focused on a family of animal lectins called galactans for many years. And last year I gave the title of glycan binding protein and their functions, and I gave a broader overview of the area of glycan binding proteins. But this year I thought I would focus on galactans. And also I thought I would focus on more recently emerged area in galactan biology. That is, inside a cell, galactan can recognize glycans. I will give you how this area developed. Unfortunately, I thought we had a problem of sharing from my side, so I need to have Tzu side advance the slide. And also, I'm not able to point using my pointer, but I think it should be okay. And I hope you will get. I think I would like to convey to you is how one area of research, meaning galactons evolve. And to me, if I would like to convey a message, I would say that always try to pursue the truth, meaning that this is how science developed. It's like in the galactic area, things are not quite clear at the beginning because the field starts with identification of molecules. Then the pursuit of function started. Then it really took many years to realize even the approach to take to understand the function of this family. So this is actually I would like to convey. And second is share with you. Through all these years research, finally, area that is to me is exciting emerged, and that is the protein family that can bind glycan, can recognize glycan becoming exposed in the cytosol. I hope my presentation will be clear in that regard. So next slide, please. Next slide. Yes, yeah. So when I talk about glycans, actually this is the very, although simplified, but I think pretty elegant way of presenting the field. And I can say that it's really not that complicated because we're dealing with glycoproteins, glycolipid, or called glycosphingolipid. It's mainly glycolipid, and the proteoglycan, and composed of protein backbone and the glucosaminoglycan. Even with regard to the sugar components, we're dealing with on the left side mainly galactose, glucose, mannose, fructose, and sialic acid. It's actually simpler than protein because just these five mainly these five sugar. And but then you see xylose and the two others. Those are components of glycosaminoglycan. And pretty much unless you work on proteoglycan, you do not need to worry too much about those. Then there's N-acetylgalactosamine, N-acetylglucosamine. You don't need to worry too much. And glycoprotein has contain both O-linked glycan and N-linked glycan. And but if I say it's really relatively simple, it's not quite true. You see this branching of O-link, both O-linked and the N-glycan, and can become pretty complicated. But for the purpose of today's talk, you really do not need to worry about this being maybe very foreign to you. And you just see this N-glycan, O-glycan as a pattern. And I pretty much, this is probably the only slide I will show this branching structure. But when I refer to glycan, you just think about a pattern. It's composed of various sugars. Okay, next slide. So then the main topic is glycan binding proteins, also known as lectins in Greek word, and also this has been known for years. But lectins is sometimes not easily understood. So I think now a better term is glycan binding protein. So this is the protein that bind glycan. And on the screen is the major types of glycan binding proteins. So selectin, I'm sure you're familiar with selectin. That is, they really put the glycobiology on the map. It's a very well recognized, very important cell tissue protein that regulate trafficking of leukocytes. And there are just three: L-selectin, E-selectin, P-selectin. So these are C-type lectin. And there are also numerous C-type lectin-like receptor, and the hepatic lectin is an example. But this is very confusing and complicated area. Professor Shiliang Xie is the one who I think is doing a lot of excellent work in this area. But if for some reason you got interest in glycobiology, be careful not to get, I hope you won't work on C-type lectin-like receptor. Is really, if you're already working on that, don't worry, but otherwise, this is a very complicated field. On the other hand, another one is SIGLAC. I talked about that. I went over this a little bit last year, and this is sialic acid binding contain immunoglobulin-like domain and lectins, SIGLAC. And very important, very interesting field. It's still wide open. There's a lot of research still to be done. Then galactins. Galactins are different from other major types of glycan binding proteins in that they do not have transmembrane domain. So they are not transmembrane protein. They do not have a so-called signal sequence or leader sequence. So they are made to be present in the cytosol. Although they can be secreted, we do not know exactly how they are secreted. Secreted through a non-classical pathway. So this makes the study of this glycan binding protein family, galactin, a little bit complicated because we need to deal with intracellular aspect and extracellular aspect. Today I will focus on intracellular, and I would maybe I'll go over several times about issues when you deal with extracellular function. Okay, next slide. So galactins, they are beta galactoside binding animal lectins with conserved carbohydrate recognition domains, CRD. They are evolutionarily highly conserved in worms like nematodes. For example, you're familiar with model organism like C. elegans, and they contain many galactins. And all mammals contain various members of galactin family. There are basically three types. One is a prototypical type who just have one CRD, but they often exist as dimer. I will talk a little bit about galactin 7 today. And tandem repeat, they have to have two CRD connected by a short peptide linker. I think I will talk about 4 and 8, 12, today. And the galactin 3, we actually discovered, we're the first group to clone and sequence galactin 3 before this family of protein is designated as galactin. And it's actually quite by accident. And it contains one CRD and non-lectin areas. At the bottom, I show this sequence comparison among different galactins. And the sequence identity is pretty low. It's only 30-40%. But their areas, they are highly conserved. These are called the signal sequence. I'm not sequencing, but almost like a signature that if you have this amino acid, they are considered as the feature of a galactin family. And this happens to be involving carbohydrate binding. But another message is, although they are relatively large family, but they are not really should be considered as a various member having redundant function because they are really not sequenced. They are quite diverse in terms of sequence. Next slide. So some galactins have wide tissue distribution, like galactin 3 we discovered is almost expressed by almost all the tissues and the cells. Others have high tissue specificity. For example, galactin 7 I will talk about, and this, especially galactin 12 is expressed mainly by adipocytes, 脂肪细胞. I mentioned already they do not have transmembrane domain and also no classical signal sequence or called leader sequence. Therefore, they are found in the cytosol, but sometimes they translocate to nucleus. They can shuffle back and forth between cytosol and nucleus. But they can be in the extracellular space, probably through a non-classical secretory pathway. We don't. There are many possibilities being published, but we don't quite understand exactly how they are secreted. So by just this description, you think these are or can be just typical intracellular mediators, and which is in some sense is true. if you know the definition of lectins, lectin is 凝集素, and if you add many lectins and plants contain a large number of different lectins, if you add those lectins to cells, the cell often aggregates. And because they are bivalent or multivalent. Chinese is nice because it's 凝集素, and it's meaning that they can cause aggregation. And so it's consistent with this protein being bivalent or multivalent with regard to carbohydrate binding activity. And you'll find that this field is relatively complicated. And but I hope toward the end I'm going to simplify the field, because the true functions start to emerge. But nevertheless, this gelatin have been implicated in the pathogenesis of a variety of diseases. Here is I would like to we need to be cautious, because if you got curious and read the literature, and if any of the paper deal with, including ours perhaps, dealing with in vitro function of gelatin and using approach that add gelatins to various cell types, then that may not be a true function of these proteins. So I hope this message will become more clear. Therefore, even though it's implicated, it's some of the you need to read, you just be cautious when you read the literature, and this will become more clear as my talk go on. Next slide, please. So gelatin 3, I mentioned that it has one CRT and along this non-lectin area, and it's from oligomers binding to multivalent carbohydrate, meaning that it becomes multivalent through this way. As I mentioned, it has a wide tissue distribution, and just like I mentioned, it's present in the cytosol and the nucleus. So although a variety of cellular responses could be easily demonstrated by using gelatins added to cells exogenously, meaning 外加的, and that's why we did this kind of experiment quite a bit early on. But then we thought it was important to elucidate the functions of endogenous gelatins, so 内源性, 内源的 gelatins, and ask whether the gelatins have intracellular functions. So as I mentioned, this is a very important point. I really hope I get this message across to you. To exaggerate this point, but which is true, I mentioned plants contain many lectins. If you take any of those plant lectins, 凝集素, and you add to human cells such as T cell, B cell, and this T cell, B cell get activated. And this is an in vitro experiment. And clearly those are not the function of those lectins, because plants do not have T cells. I hope, but this is the point. So I can say with any lectins, whether it's animal lectins or plant lectins, if you add these lectins extracellularly, exogenously, and see the response, those may not represent the true function of these family proteins. Okay, but I admit very early on we didn't know much about this biology, and that's how we pursued. But when we become more and more clear about the various issues, and then we start to adjust our approach. Next slide. And so we started to address the endogenous gelatin, function of endogenous gelatins, and whether they have intracellular function. And fortunately, we started to discover various functions. I won't go through this one by one, but this is actually, in some sense, it's just basically my career spanned over about 20 years. The first one was gelatin 3 has anti-apoptotic function. It's interact with a well-known apoptosis regulator, BCL2. It's actually bind to BCL2. And then we found its role in phagocytosis, T cell response, and so on. I won't read this. And next slide. So this involve interaction with intracellular regulators, for example, BCL2. Another intracellular regulator is now pretty famous called ALIX, ALIX, at the upper right-hand corner. These are independent of glycan binding. BCL2 is not glycoprotein. ALIX is not glycoprotein. However, carbohydrate binding domain might be involved, and this applies to binding to BCL2, which we have not fully established this point. But CRD might be involved, but not because they bind glycan, just because that site fit some peptides contained in BCL2. But the binding to ALIX is different. Binding ALIX is the non-lectin part of galactin-3 bind the ALIX, so it does not involve CRD. Next slide. So then we have been studying other galactins and elucidate their functions. So I'm going to give you some examples. Next slide. So galectin 7. I mentioned some galactins have a more restricted tissue distribution. So galectin 7, as an example, is expressing stratified epithelial cells like skin, esophagus, tongue. They are stratified epithelial cells, highly induced by 53. Galectin 3, I mentioned, is anti-apoptotic, and galectin 7 is pro-apoptotic. And we show its tumor suppressor. These are not really key points that I want to talk about today. And they also bind BCL2. And if you look at the right side, you find that this is the staining of galectin 7, either in transformed or immortalized skin cells, HKT, or HeLa cell transfected with galectin 7, and the green fluorescence is the galectin 7, red is nucleus. So you see this highly enriched nuclei. And that really helped the galectin field because people start realize this can be intracellular. And the people even say galectin 7 is intracellular galactin, which is not exactly right because all the galactins are intracellular. Just some of them get secreted, and some of them, they are more studied of how they work if you add exogenously to cells. Next slide. So we started to focus on the role of galectin 7 in skin keratinocytes because I also actually dermatologist, and I try to. We discovered the galectin family before I became a dermatologist. So after a while, we started to want to study more functional galactins in skin, either normal skin or skin diseases. And then we found that galectin 7 through this pathway on the right side is suppress keratinocyte proliferation. So in the absence of galectin 7, or when galectin 7 is downregulated, a cell proliferate more. This is published a few years ago. Next slide. Then we try to link this to inflammation. So then we found in a previous slide, we showed galectin 7 upregulate microRNA 203. But here we show galectin 7 downregulate microRNA 146A. And through this is suppress this MAP kinase pathway and end up suppressing IL-6, IL-8 production. So when galectin 7 is downregulated, there are more IL-6, IL-8 production. Next slide. So I say that when galectin 7 is downregulated, because that's how we found that's the case in psoriasis, 干癣, a skin disease, very important skin disease. And the galectin 7 level compared to normal skin is lower. And then so the question was, what happened to the keratinocytes when galectin 7 level is suppressed? And we link to keratinocyte proliferation, I mentioned. We link to cytokine production. And so this is a summary of that in a cartoon fashion. And it was published in Journal of Clinical Investigation just last year. Next slide. Sorry, I went through this very quickly because this is really not a major focus of my talk. Then galectin 12 is very interesting. It's contained two domains. And N-terminal domain is homologous to other galectin CRD. Even that, we're talking about 30-40% of sequence identity. The C-terminal part is quite divergent. Almost you cannot recognize as a galectin family, but they still some amino acids they're conserved. And I mentioned that there's more glycan 12 has a more restricted tissue distribution. It's mainly expressed by adipocytes. And we published a paper about this function adipocyte. So this also brings up a challenge here to our group. That is, by studying a family protein, we continue need to learn something new. For example, by the immunologist, we had to learn about adipocytes. But fortunately, this I think, as you, I'm sure you agree that collaboration is important, and we always try to engage experts in various fields. And in this case, every expert in adipocytes. Also, I have very talented postdoctoral fellow who learned about adipocytes, about this adipose tissue very quickly, and we have been productive. So next slide. And in terms of the function, it turned out glycan 12 is localized on lipid droplets, and it suppresses lipolysis. So lipolysis, the degradation of triglyceride to glycerol and fatty acids, it suppresses this. So we generate knockout mice, and knockout mice actually thinner. They have had less fat tissue. And our working model is because glycan 12 is suppressed lipolysis. When it's absent, there's more lipolysis, and therefore there's less triglyceride in the cells, smaller lipid droplet, and smaller adipocytes. Yeah, and so yes, so next slide. Okay, so I mentioned that you can easily show in vitro experiment glycans can cause some cellular response when you add glycans to extracellular space exogenously. And I mentioned that this might not be their physiological or pathological function or in vivo function. I guess I'm not quite sure I mentioned why, but one of it is specificity, because glycans, especially glycan 1 and 3, they bind numerous glycoproteins. You really cannot predict in vivo when glycans are released, where they go to. So even though you show that it does something to T cell, we really cannot predict it's going to even encounter T cells. But I'm not quite sure it's entirely clear. But don't worry about that point. Just say that just recognize there's a question mark in vivo. Okay. And then I share with you our work on glycan 3, glycan 7, glycan 12. And we ourselves is more and more convinced that the glycans work inside the cell through protein-protein interactions. And I think the field appreciate that, and also recognize they have glycan work outside the cell, inside the cell. And inside the cell, our group is often recognized as the group that kind of started this direction of research and have published quite a few papers. But then so while we're doing progressing in the study of function, we continue to ask, these are glycan binding proteins, and they should bind glycan. They do bind glycan outside the cell, but inside the cell, do they function by binding to glycans? Okay, next slide. So before that, because today's talk, the title is intracellular immunity, I need to introduce a couple of key players, and these are many of you already familiar with, maybe more so than myself. But I just thought I just very briefly mention, happy to show you 2 cartoons. Next slide. So one is autophagy. Autophagy early on was discovered to be a cellular process that take care of damaged organelle or damaged proteins. Meaning phage is engulf, and auto means self. And but it's also known to be important in recognizing intracellular microorganisms and so on. So, for example, on this cartoon, if a microbe got inside the cell and they escape, initially it's in phagosome, but they escape goes to cytosol, and the machinery quickly recognize this being danger signal and then take care of that by autophagy. On the right side, you see the major components of autophagic activation, including P62, I will mention NDP52 at the bottom, and LC3. Okay, next slide. And this another one is inflammasome. I'm sure you're familiar with this. So in the middle, there are fatty acids, ceramide, and so on that can activate intracellular NLPR3, a major component of inflammasome. And this NLRP3 start oligomerize, engage another component called ASC, and eventually caspase-1. And the caspase-1 get activated, become active caspase-1, and that then convert pro-IL-1 beta and pro-IL-1 IL-16 to active IL-1 beta, IL-1 16. This get 18, this gets secreted, and this induce inflammation. In the meantime, caspase-1 also degrade another component called gastromin D, and gastromin D get cleaved and induce pyrotosis. So this inflammatory activation result in pyrotosis. One is cell death. The second is inflammatory response, like induced by IL-1 beta and IL-18. I'm sure that this whole thing is not as simple as shown on cartoon, but this actually illustrates the key components. Okay, next slide. Yeah. So now the question is, do glycans bind glycans inside the cell? By glycan, I meant this glycoprotein, glycolipid, proteoglycan. And as I promised, that so far I have not said anything about this very complicated branching structure, and that's continued to be the case. Just think this as a pattern, right? N glycan, O glycan, and the inside the cell there's a very important the glycosylation is O-linked N-acetylglucosamine on proteins. Many signaling molecules, as you know, that get phosphorylated on serine and threonine. They can also be glycosylated. We call the N-glucanac, O-glucanac, O-N-acetylglucosamine. This monosaccharide, the glucose, is N-acetylated. It's also bind to serine and threonine. In fact, the site of phosphorylation can also be site of glycosylation, and they can be interconverted. And I think this is a very important intracellular signaling mechanism, but let's not. I won't talk about that. But I just say that the glycans do not bind this O-glucanac. So this is not the answer. So they do not bind. Normal situation, all the glycan I show on this slide. Next slide. Next slide. So because this glycoprotein and glycolipid, their synthesis initiated in the endoplasmic reticulum and goes to Golgi apparatus and go to post-Golgi network, secretory vesicle, and get exposed, echocytosed. They can be sometime under some conditions internalized and to endosome and degrading lysosome, or they can be recycled back to extracellular space. And just like when I mentioned, galactans do not have signal sequence, and so they do not go through this pathway. And so galactans are cytosolic, and those glycans I talk about, glycoprotein, glycolipid, they are in the lumen of this organelle. Endoplasmic reticulum, Golgi, endosome, lysosome, they're in the lumen. Galactans in the cytosol. So the answer should be no. That's what I thought very early on. I said that they don't bind each other. But I remember back then I kept thinking about under what condition there might be chance, and I have some ideas over the years, but I missed something very trivial. Next slide, please. Because I mentioned that I'm actually Weisstein Shea mentioned that I'm a chemist, and later on I switched to immunology, but I never took an immunology course. I never took a cell biology course. I didn't know this is a very important process occur inside the cell. That is endosome or lysosome. can be damaged. So I say endolysosome, actually no such thing as endolysosome. It's endosome or lysosome, because they are very disruptive material. I'll come back to at the end, what are these disruptive material. But anyway, there's various disruptive material can get inside the cell. When they get inside the cell, this either endosome or lysosome can be damaged. And I mentioned that these glycans I talk about, glycoprotein, glycolipid, they're in the lumen. But when this organelles damaged or this vacuole get damaged, this glycan represented by this triangle, a Y-shaped structure, which is oversimplification of what actually their exact structure. But anyway, then galactans, usually they are present in the cytosol. If they are present in the cytosol, they're present in sufficient amount. They don't need to be induced. And then they can quickly recognize this exposed glycan, glycoprotein, glycolipid. Recognition is one thing, but they can lead to downstream event such as autophagy. I'll get back to this point soon. So this process is as I said, I really didn't think this occurs. And after I knew that our lab started to catch up, and I started to be more and more amazed by this situation that is galactan abundantly present in the cytosol. They actually have a chance to function through binding to glycan. Although normally there's no, the two galactans and glycan, they don't encounter each other. Next slide. So early on, so Eugene is my graduate student at UC Davis, and after 5 years or so, she accumulated a lot of data, and she graduated without a paper. Then she came back to Taiwan. I returned to Taiwan before that, and she came back and joined. Also continued to work on this project in my lab in Taiwan at Academia Sinica. And the work we work on Listeria monocytogenes, which is a very commonly studied bacteria that invade macrophages, and so they get phagocytosis, so they invade and they confine in phagosome, but they want to escape by secreting something called LLO, Listeria lysine O. They cause vacuolysis and they escape, come out. Okay, so next slide. So very early on, Eugene, this is a confocal time-lapse study, and with galactan labeled with the green fluorescent protein, and Listeria is labeled with the red fluorescent marker, and this is a monocyte. So I think you can see the red signal. Those are Listeria. The escape takes place about 1 hour or so. So when you see between around 73, 78 minutes, you see the red fluorescence still there, but you start to see green fluorescence accumulate. And certainly between 78 minutes, 79 minutes, there's apparently a large amount of galactans 3 accumulates. And we were quite excited about this finding. Next slide. Next slide. Yeah. Next slide. Yeah. So then a very important paper came out. This is actually very exciting. Even though I say that we wish we'd be the one to publish a paper like this, but nevertheless, I thought this is very important. This is very exciting, and I think the citation must have exceeded 1,000 and keep going up. So what they use Salmonella, they use Listeria. We use Listeria. So Salmonella get inside the cell. They have phagocytosis, so they reside in the phagosome. But they also want to escape, and there's a loss of membrane integrity because they secrete some substance. Then this intraluminal glycan get exposed to the cytosol, and they show that galactans 3, 8, and 9 bind to this glycan, exposed glycan. They actually bind to N-glycan. Furthermore, I think this is the most important part of this paper is galactan 8 can bind NDP52 I talk about. So galactan 8, one CRD bind glycan. Another CRT bind ND52. This just nobody would predict that this interaction occur, but that's actually that's how they demonstrated. Then autophagy take place, and autophagy engulf salmonella. They try to escape, but now they are phagocyte. They get the autophagosome, confined in autophagosome. Okay, next slide. After we saw this, we knew that this really should be the research direction. Early on, we were more focusing on glycan-3 binding to Listeria bacteria, not really think about host glycan. But we modify our direction very quickly, and then we show that glycan-3, so opposite to glycan-8, is actually suppress autophagy. But I must say that they work on Salmonella, we work on Listeria. We didn't show in Listeria glycan-3 suppress. Glycan-3 suppress, but we didn't show that glycan-8 promote. In Salmonella, we do not know whether glycan-3 suppress autophagy or not. But anyway, this is how we concluded. Next slide. So then we paper published quite a few years later after the Nature paper, but we were very pleased with this paper, and took Yijin quite a few years to complete, but this was selected as a choice. So glycan-3 protects intracellular Listeria by suppressing autophagic response in a host and glycan dependent manner. So it's very clear. And then 3 and 8 antagonize each other in antibacterial autophagy. I take back, actually, for Listeria, we did show these 3 antagonize, but I say that in Salmonella, I don't know whether it exists. But anyway, so this is our conclusion. Next slide. I'm going to skip the next couple slides because I need. So these are a little bit more tedious, and I'm not sure it's worth talking about this. This is the key message of the paper we published, because we knew that we need to include more information, and this is actually it's very, very interesting, the conclusion. But next slide. Next slide. Yeah. Next slide. But anyway, the conclusion is that we can modify the cell surface glycosylation. It can be done easily. This also occur under various conditions. So cell surface glycosylation can be modified. Then the cell surface glycan get internalized. Then when the endosome lysosome get damaged, they get recognized by glycans. So even if modification occur on the cell surface, the response can be altered. This can result in different cellular response, not because cell surface modification, what happened on the cell surface per se, but because the change get recognized inside the cell, because glycan-3 and 8 bind to this. They bind to glycans, but they sense this alteration and lead to a different differential response. I'm not sure this is clear, but if not, don't worry. We need to move on. So next slide. Next slide. Yeah. So another project was carried out by Fang Yan Li. He graduate student at NTU, and this his thesis. So we work with H. pylori. H. pylori is not the intracellular bacteria, but you can secrete substances such as HAG-A, VAC-A, and then this can affect cytosolic intracellular compartment. For example, VAC-A, vacuolating cytotoxin A, can cause formation of vacuoles and can induce autophagy and apoptosis. These keywords attracted us. We think VAC-A is relevant in the study we are pursuing. Next slide. Next slide. So Fang Yan, the previous one. So gastric epithelial cells. Gastric epithelial cell, we treat them with H. pylori, and then we are really very good at looking at accumulation of lectins. Basically, this is puncta. No, you see puncta. I'm not sure. I'm quite sure you can see. So we increase the number of puncta So it's increased with the hours of incubation, and also increased with the amount of bacteria the cell exposed to. I hope you can see those bright dots in the cytosol. Yeah, those are the puncta, and those are aggregation of galectin 8. Okay, next slide. So this localized in lysosome. Previous one is localized in phagosome, and then become autophagosome. But this is lysosome because the green signal is galectin 8. LAMP1 is a marker for lysosome, and on the merge you see a white signal that is the galectin 8 and the LAMP1 co-localized. So these are localized in lysosome. Okay, next slide. So this is the conclusion of the paper. This was also published in Glycobiology, also at the editor choice. So VEC-A without the bacteria getting inside the cell, VEC-A secreted by H. pylori cause damage to lysosome. And so again, glycan get exposed, and galectin 8 get accumulated. And then just like in the case of Salmonella, probably Listeria, galectin 8 trigger autophagic induction. So we add a little bit more twist to this story. That is autophagic activation induce RS, reactive oxygen species. RS is known to cause damage to organelle, in his case lysosome. So there's more damage as more galectin 8 accumulation, as more autophagy. Okay, so H. pylori can induce autophagic induction through VEC-A and through its causing lysosome damage. Next slide. Next slide, please. So I'm going to go through the next few slides very quickly, and it's basically we got excited about this model, so we use a model system using photosensitizer, and next slide. Next slide. Yeah, you see this puncta, and stay here. So you see, go back to last slide. So this is galectin 3. You see the puncta, and this the photosensitizer get endocytosed, and then when you shine red light to the spot, it causes the endosome to be ruptured, and then galectin 3 accumulates. So the green dot you see is the galectin 3 GFP fusion protein, but that place is where photosensitizer locates in endosome. But then when you illuminate that area, the endosome get damaged and galectin 3 accumulate. And the previous slide was we show is we use the laser to irradiate the spot, but this one just using the red light equipped with the microscope. Yeah. Anyway, okay. Next slide. Next. Yeah. So this one we use super resolution, show galectin 3 and 8 localized in different microdomain, but. And this just shows that autophagy adaptive protein PCC2, LC3 I mentioned, they co-localize with galectins. So this means this place eventually is also being becoming autophagosome. Yeah. Next slide. Next slide. Yeah. Next slide. Next. Yeah, next slide. Okay. So why we think this is important? So there are many substances cause endosome damage, endosome or lysosome damage. Intracellular pathogens, I talk about Listeria, Salmonella, but also bacteria forming proteins like VEC-A. And the virus can cause this too, but it has to be non-envelope virus. And the protein aggregates such as those responsible for neurodegenerative disease, neurological disorders. So next slide. Okay, so Chen Yuzhuang she got interested in gelatin, and she worked on Huntington disease, and she got attracted to the idea of gelatin-3 can cause organelle damage, and wonder about its relationship to disease like Huntington. So Huntington disease, the mutant Huntington in microglia is important for the disease. And the protein is aggregates is known to, so mutant Huntington aggregates inside the cell. So mutant protein aggregates can cause endolysosomal damage. It's known, and she showed that. And then she confirmed that gelatin-3 accumulates around damaged lysosome. It co-localizes with LMP1. So on the right upper corner, you see gelatin-3 structure. You see the red dots. Those are LMP1, and usually this is lysosome is damaged, and the gelatin-3 get accumulated. Also, she showed that lysosomal rupture can cause inflammasome activation, and gelatin-3 enhance this inflammasome activation, and end up having more I1 beta secretion from these microglia cells. And she proposed this is the cause of neurodegeneration. So this is to me, it's a very exciting story, and was published in Nature Communication just about 2 years ago. Next slide, please. Next slide. Yeah, so protein aggregates, and another example is particulates. I'm sure you're familiar with nanoparticles being used a lot in delivering the drugs, but we also exposed almost daily like particulate matters, and these particulates can cause intraorganelle damage also. And this is actually gelatin-8 now is being used to show that nanoparticle can go from endosome to cytosol, and those nanoparticles need to go to lysosome to deliver the cargo. And so, but now we know that by doing so, actually you cause something happen. The something is gelatin get accumulated, then cause some inflammasome activation, autophagy response. So it's not inert, but I'm not sure how big the effect is. But it's something those working with nanoparticle need to be aware of. Then another important substance is not really substance, but the process is cellular stress, such as oxidative stress. I already mentioned that earlier is ROS from autophagy cause lysosome damage. So it's one example, but there are many oxidative stress, different various forms. The ROS is pretty commonly studied, and that can cause lysosome to be damaged. So again, I in case it's still not quite clear, so various disruptive materials can get inside the cell through endocytosis or phagocytosis, and initially, so then the glycoprotein, glycolipid go along with these disruptive materials and become part of endosome, phagosome. But these glycoprotein, glycolipid, they reside in the lumen. But then this disruptive material can cause damage to these organelles. Then gelatins get attracted to the sites. And they lead to, through various mechanisms, autophagic activation or inflammatory activation. So I believe I talk about novel mechanism mediated by cytosolic exposed glycans, and this can lead to diverse cellular response and innate immunity and inflammation, and relate to disease. So next slide. So again, I come back to this extracellular function. Although if you go to literature, you'll find numerous examples of what happens when you add gelatin to cell. And but we really need to think whether those are in vivo function or not. Inside the cell, I think I believe personally that they do work inside the cell through protein-protein interaction. But maybe more importantly, is function as a very unique protein family to recognize glycan. So it's intracellular glycan sensing molecules. I think I pretty much used up my time. I hope to leave some time for questions, so I'm going to skip the next couple of slides. Could you show me what's the next slide? Next slide. Yeah, go back to the previous slide. Okay, so I'm going to skip maybe share with you those are more recently published and or not published. I will share with you maybe next time. So thank you for your time. I'm sorry for the not being able to control slide from my side.
Thank you, Professor Liu, give us the wonderful talk. So, have anyone raise your hand to ask the question, or someone in the on-site want to ask the question?那我先问好了。
OK， 对，用中文都可以，不一定要英文。对。
刘教授，院士，我记得您前面有讲到 galectin 3跟 8在有一些 system 是一个 antagonize each other， 对不对？
是。
所以目前有关于在 lysosome 这个的部分，你们现在好像还是是 focus 在 galectin 3嘛，对吧？
我们 8也做不少了，对。
所以它也有类似的 antagonize 的作用吗？因为我觉得这上
对，谢谢你这个问题。我们就回到那个 H. pylori 的话，因为 H. pylori 说 8， 然后并没有做 3， 然后我现在有一个博后在做 3， 我觉得我们可以把3跟8比较一下。那目前为止没有看到它是 antagonize， 就是在同样是 VEC A，就是 induce lysosome damage， 然后造成 autophagy。他看到3跟8都是在同一个 direction， 所以那个很可能只是 listeria， 所以我想我们这个在 H. pylori infection 情况下，我们最后做的结论是会把这个更 clarify， 对。
那如果它本身两个没有 antagonize 的作用，因为我记得您在去年的 talk 里面讲到那个 antagonize 的情形，我觉得是非常 amazing。那今年这个部分，我会想到另外一个想法，就是说，如果他们没有就是没有 antagonize each other， 那他们会不会有类似？有时候我们在 pharmacology 或是 physiology 里面，我们会有类似所谓的 facilitate， 或者是这个 add on 的作用。好，因为我知道院士你们这个一些相关的研究都非常的新，而且也很超前，但是我们也会很好奇，我个人会很好奇的说，尤其是现在我们过去都会 focus 在 protein interaction， 那您这个部分是这个glyco protein 这一块的，或者是说 glycan 的这一块的，他们也会不会有像我们以前在想的，就是会有一些 interaction？ 所以这个也是想请问一下刘院士，你们现在的一个想法。
对，谢谢。那个刚才比较快的跳过是发现就是 galectin 3跟 galectin 8在不同的 micro domain， 因为它是 bind 不同的，所以虽然我说早期我说他们没有 specificity， 但是他们有 selectivity， 说 3 bind 的那个我们这个 glycan array 都有些，不是我们做，就是别的团队做出来 glycan array， 可以看得出来 3 bind 一群 glycan， 8 bind 一群不同的，有些 overlap。那这个比如说是我刚刚有个重点，我也很快也跳过去了，就是如果是一个 glycan 是 silylated 的话 ，glycan 3 不 bind， 但 glycan 8有 bind， 所以他们就是有 selectivity。那我们用那个 super resolution， 我看到3跟8是 bind 不同的 domain， 所以他们之间不 compete。所以我们如果说它 antagonize 的话，为什么 antagonize？ 我想是 downstream， 就是 autophagy 那个的那一部分。一个是 bind NDP52， 然后 induce， 一个的话 也许 retard 这个 process， 对，不是这个 glycan recognition 这样。那我们现在觉得这个你问的问题，我们真的是很关心，就是应该在不同的 system， 比如说 H. pylori， 把它搞清楚，或者 Salmonella， 我们再回去做看看 3 跟 8 的关系是什么。所以这个我想 clarify。那如果说是两个不 antagonize 的话，3 跟8应该是 additive 了，至少 additive， 对，或是说是也许可以讲说是8比较强，目前为止8比较强，它是 binding 也快。bind 得非常快，我刚才就是可惜时间关系，那个 model system 我用 photosensitizer， 那个 3 的话要一阵子它才 accumulate， 的话就是几乎是我们一照了光，还来不及照相，它已经在那里了。对，就 0 minute 就看到很聚集很强的，所以 8 是非常快。那 3 的话比较慢，所以也许3也许并没有那么大作用，也不一定。所以要看 additive， 也许看不太出来，也不一定。不过这个总是很重要的question， 然后再加上galectin 4是不是也会有影响？那 galectin 9这些都值得去看。那这些之间的关系，我想我们也是很有兴趣的，对。
OK， 我们有一位那个学生，应该也是去年问您的那位学生， 我们早上有在聊的。
OK， 嗨，
可以给他那个 microphone， yeah。
Thank you, Professor, for the amazing speech. I have a question. My question is in two parts. So the first one is about the role of enzymatic removal of sialic acid. That is a sialation, and because it modifies the function of galectins. So in the paper that you showed, that's on slide 45 in Huntington disease, where GAL3 was shown to be implicated in the pathogenesis of Huntington disease. Was there anything done with maybe the enzymatic removal of sialic acid from the cell surface, or maybe internally as well? Does it play any role in Huntington disease pathology?
Yes, yeah, thank you. Very, very good question. So we have not done that. But it's giving me an opportunity to share with you. I also share with VP Chair, and so this area becomes so interesting, and investigators at another Institute of Biological Chemistry, got interested in this whole concept of galectin-glycan interaction. And then they form a team to tackle this. They got funding. And so we already have many meetings. And I kept learning something new. And what's new is I talk about the modification on cell surface silation. But during the meeting, it then became clear to us that those exposed glycan can be modified intracellularly, just like you mentioned. So you must be familiar with neuraminidase, 1, 2, 3, 4. And they are cytosol, they can be in mitochondria. And so I believe what happened, what I described intracellularly when the endosome got ruptured and the glycan get exposed, that this picture might be oversimplification. And I'm sure they get modified. And then there will be competition of binding and the modification. Yeah, and this can be either modified or this, or even if you consider this as a way of treatment, maybe you can just modify this process. But I believe this occurs naturally. That is, it's not going to be just a very simple exposure and galectin binding. This neuraminidase might get activated, may get engaged, and cleave cyclic acid. When that occur, then this means that there will be more galectin-3 binding, less galectin-8 binding. Yeah, thank you. This is very, very good question.
Just check the paper to find out. I wanted to ask the model, the genetic model of the mice that was used, and maybe the expansion of the CAG gene that for the mutant autentin protein in the... Yes. So I wanted to ask, but I will just check the paper and find out that.
Okay. Yeah.
Thank you.
All right. Thanks. Yeah, you're talking about the polyglutamine, right? Polyq.
Yeah.
Polyq. Right.
Because I know the Professor Yi Zhuangchen, she always used like R62 mice and R61 mice.
Right.
It's the Huntington disease model.
Yes.
So actually, you can check the Nature Communication 2019 the paper. Okay, yeah. 那我们现场或者是那个线上的老师同学，如果你有问题的话，也可以，如果是在线上的，可以打在那个 note 上面，然后我们也可以就是代为发问我们刘院士，好不好？不晓得我们在座有没有哪一位想要再问相关的？我要 cue 那个我们那个黄卫汉医师，
中文问好了。
好，黄医师你好。
刘院士好，因为其实之前在台大跟田文芬教授还有周文坚教授的时候，就对于当年的那个。就是在 AML 里面，我们去看，就是那个时候，我记得老师们有跟刘院士合作，就是在 AML 的部分去看，就是 encoding 感听税的这些 gene， 它其实是 overexpression 的情况。那这几年其实不管在 hematological malignancy 或 solid cancer， 其实都可以看得到感听税在这些 cancer， 不管是在 adhesion 上，或者是它 disseminate 上面的这个角色。那其实这几年甚至有跟它对应的这个 lecture 有相当多的这些 paper 陆续在出来，那想请教刘院士这方面的这个 comment， 就是说，因为我实在很好奇，说现在也有这种 treatment 好像慢慢要浮现出来，只是好像都还在很前期的部分，那不知道院士的看法。
对，就是glycan 跟 cancer 关系是非常多的文章，然后也就变成可能是个 target for therapy。那里的话，就是 association 是蛮确定的，就是它的 expression level 啦，然后有一些evidence。那所以是有几个公司在 pursue。那边有一个可惜的地方是，就是早期的时候没有搞清楚是它这个 endogenous protein 的功能，比如说你在 cancer 里面有增加了。那它怎么样子让这个变成它是一个 oncogene？ 对，它可能是 tumor suppressor，glycan 3大概应该是 oncogene，glycan 7也是 tumor suppressor， 它是怎么作用的？那如果是 oncogene 的话，要 inhibit 它，那他们就会用有一些会 bind glycan 的东西。那有时候是大分子，比如说非常出名的是这个modified citrus pectin NCP。那这个在20-30年前他就说是它是 bind glycan 3， 那这个那个公司后来做也没有把它搞清楚，就是继续用这个，或是它的 derivative， 好像有时候有效果，但是我觉得它的效果很可能不是 bind glycan 3， 而是它本身就是 modified citrus pectin 有些作用，对，那所以这边的话就变成真的是要 put a question mark。就是应该是他的 approach 没有用对，所以我一直在强调这个就是要了解。我曾经常常有用一个 sentence， 就是我们要做 precision medicine 的话，一定要 depends on precise science。对，那个话就是早期的用modified citrus pectin 是误导了这个领域。那很多人后来也都知道说这个是不对的，就是这个它就是没有办法 prove glycan 3的功能。那这个后来就变成药在用，那好像还是有一些已经在做 clinical trial 了，那这有点令人担心的。但是所以这是 cancer， 那 cancer 的话，事实上是真的是蛮多opportunity， 然后但是不是最好的 target 目前为止还不能讲。我们自己手上也有一些，比如说跟 colon cancer 的关系，那我们就比较拉高跟 Y type 老鼠，看到它有差别。那在 3 啦这些都有，那是不是最好 target 不确定，那这都要靠大家来做。然后到了 challenge 的地方，就是你知道它是一个 target 的话，那它是用什么 inhibitor？ 那这个的话，就是因为过去是用比如说 modify switch spec 的那些是不是 specific 的，然后真的是没有搞清楚它的 mechanism 它在哪里作用都不清楚。但是如果真的要 specific inhibitor 的话，不是那么容易。但有个公司做得很不错，它很早期就做一直在做 compound， 然后它有蛮有名的叫做 TD139， 然后这个他们在 clinical 在做，这个不是 cancer 了，是 idiopathic pulmonary fibrosis， 证明在 phase 1、phase 2有效。这个在这个可能有用。那即使那个的话，对不是完全是 galectin 3 specific， 有点有的时候也会弯了什么的，对，但是我觉得已经相当不错了，所以这个是我们会陆续看到，这个来看是不是可以在如果是 galectin 3是在某种 leukemia 是有影响的话，是不是可以用这个？那是不是可以用 TD139？ 那我们这边的是跟生化所林俊宏老师合作，他在做不同的类似 TD139 的那目前也找到一些，然后我们也在看在 leukemia 上面是不是有影响，就是 in vitro 了。这些我们也都觉得说它一定要进到细胞里面才会有用，对，那是目前初步看到一些效果。所以目前事实上是我不太确定除了那个 TD139 之外，有没有其他的公司在 on the right track？ 对，谢谢。那至于 cancer 的话，是哪一些是可能都有蛮多，就是 correlation。比如说我们的也是跟我合作的陈一端老师，他现在是对那个diffuse large cell B cell lymphoma 是对也看到 galectin 3那没有他看到是 galectin 8， 看到 galectin 8的影响，对，所以总之一直是一个可能性，还没有比较 challenging 的，但是我想不晓得当初你们后来有没有看到什么correlation 觉得说是可以当 target。
因为我自己做的是比较偏 multiple myeloma 那个部分， 那 myeloma 上面也是跟 galectin 3比较有关。
OK， 对，都可以再去看是不是它是不是 target。对，那这个我们tool越来越多，所以那个已经是好多年前了，对，是可以再 revisit。对，所以你那个时候就毕业了，然后到实际来，是吧？对，恭喜。
好的，那我们的时间也差不多了，那我们是不是再一次的谢谢我们刘院士给我们这么精彩的演讲，